To me, the most important thing in our industry, and you'll hear this throughout the whole presentation when you're asked about policy or manufacturing or cell therapy, is patients are first. And right now in the environment that we're in, patients are a little bit out there in the atmosphere, floating around wondering when they're going to be first again. We have to come back to patients first in our industry. My journey into biotech, I won't go all the way back to 1960 when I was born, I promise you. But 1973, when I was 13 years old, I was diagnosed with severe Crohn's disease. That colored my choice to go to College of Pharmacy and become a pharmacist. When I went to pharmacy school, my second year in pharmacy school, my Crohn's acted up, and I went from being somebody with a colon to somebody without a colon. So I had my colon removed, I ended up with ostomy surgery, and when I woke up from that surgery, I was a different person. I was alive, I was hungry, I was really excited to be alive, and I decided at that point in time that rather than be in the business of practicing pharmacy, I wanted to be in the business of discovering, developing, and the business of the pharmaceutical industry. Nkarta is an autoimmune company. We're focused on a number of different autoimmune diseases. And we're treating these autoimmune diseases with engineered, off-the-shelf, allogeneic, on-demand, CD19 targeted natural killer cells. Natural killer cells are a cell that your body makes naturally, so we use healthy donor-derived blood to isolate NK cells, and we expand them and engineer them so that by the time these cells get to the patient, they're in a vial, they're ready to be dosed. The patient doesn't have to go through apheresis. We decided to work in autoimmune disease after having started in cancer. Natural killer cells tend to be short-lived, and so the long-term expansion you see with T cells don't happen with NK cells. So an NK cell is kind of a handmade therapeutic for autoimmune disease, because what you need in autoimmune disease is a very quick reset of the B cells, depletion of the bad B cells, reset to good B cells. Those patients, then you want them to have durable long-term responses. You want to have the ability to retreat those patients if they have a flare. So this is where natural killer cells are engineered, if you will, for this disease area. So we're in lupus nephritis, systemic lupus erythematosus, myositis, myasthenia gravis, scleroderma, and vasculitis.
Is that because you're seeing that common mechanism of autoimmune disease being B-cell driven?
Absolutely.
That you can...
And there are many, and what's interesting, there are many more diseases that are also B-cell driven in the autoimmune space. And the real trick in cell therapy is to make sure that we are finding one of these B-cell diseases that needs to be treated, that we could treat and potentially go into an accelerated approval, single-arm trial, pivotal trial, after doing a dose escalation.
You mentioned that NK cells, we all produce them. Everyone in this room has NK cells. Can you talk to me a bit about how just the donor supply chain works? What makes a good donor versus an inappropriate donor and things like that?
Well, the companies that do the donor selection, and there's many across the United States and across the globe, actually, they use the same donors for cell transplantation, et cetera, et cetera. So there's a network of donor centers that are built across the country. So it's really easy to use donor-derived cells. The healthy volunteers go into these donor centers, they donate their natural killer cells. They have to meet certain criteria for our manufacturing process, and they have to pass certain tests. And then they get shipped to us, and then we make these, like I said, allogeneic, off-the-shelf, on-demand, ready-to-use natural killer cells. So the nice thing about a healthy human donor is that what you're producing is a real natural killer cell that's been isolated from blood. You're not trying to make a cell look like a natural killer cell by making it from iPSCs. You're actually using a natural killer cell that's a real natural killer cell and then engineering that.
What's the data that you've been seeing so far that you can share?
We did some extensive studies in oncology, and what we found was we saw that B-cell depletion, we saw that B-cell reset. You can look at all the other CAR T data that you're seeing out there, and you go back to looking at what Jörg Schett did in Germany when he treated the first young autoimmune patients with CAR T. There are dramatic responses. They're deep, they're durable. And so their difference is cell therapy has shown a lot of promise here in autoimmune disease, as well as in oncology.
Definitely, and you've hinted a few times about your Nkarta's work in oncology, and you've kind of had that encouraging data, and then pivoting to autoimmune diseases. Can you kind of talk us through why you made that decision, why you think you're set apart from the field in this area?
Any decision that requires you to go from one patient population to another is not always an easy decision to make, right? Because if you feel like you're having an impact in one area, you'd hate to give that up to go into another area. The field was crowded in oncology 5-6 years ago, and now it's shifted to being crowded in autoimmune disease, while there's still oncology work going on with CAR T. And I think CAR T, whether autologous or allogeneic CAR T, is very exquisitely set up to be great treatment for oncology. You're also seeing great data with CAR Ts in autoimmune disease. The problem there is when you inject T cells, even a small amount of T cells, they just expand. And when they expand, they release cytokines. So they get cytokine release syndrome. They also get neurotoxicities associated with the release of those cytokines. So there's a lot of toxicity associated with CAR T theoretically from the long-term expansion. So one way to overcome that is by using a cell that has a short-lived residence time, and that's what an NK cell is.